Continuing our discussion of biological systems, we're going to go into the digestive system in Lecture 9 of this module. As usual, we're going to start off by talking about the anatomy of the digestive system. Below is a diagram listing out a general overview of what constitutes the digestive system. There are different types of digestion, starting first with intracellular digestion. This involves the oxidation of glucose and fatty acids to make energy. As the name states, this happens within the cells. However, certain types of digestion occur on the outside of cells, which is where extracellular digestion occurs. This occurs in the lumen of the alimentary canal. Mechanical and chemical digestion are both necessary to break down larger molecules into usable forms. Mechanical digestion constitutes physical breakdown of larger compounds to smaller ones. The most common example you'll see of this is chewing, or mastication as it is known in scientific terms. Chemical digestion involves enzymatic cleavages of chemical bonds within food. The pathway of the digestive tract is as follows. Starting first in your oral cavity, it travels to your pharynx, to the esophagus, to the stomach, to the small intestines, to the large intestine, and to the rectum. From there, any sort of undigested waste is defecated. We'll start with the first two portions of digestion, which involves ingestion and digestion itself. The first stop for digestion is the oral cavity. As stated earlier, chewing occurs here. This is where mechanical breakdown happens, but there is some chemical digestion in the form of salivary amylase and lipase. The pharynx connects the mouth and the nasal cavity to the esophagus, and last, the esophagus carries food to the stomach using peristalsis, which is a systematic contraction of the smooth muscles down this tract. Note that the entire digestive system is controlled by the enteric nervous system. The stomach itself produces four different types of cells. Mucus cells produce bicarbonate-rich mucus to protect the stomach lining. Stomach acid is very strong, and if it were to leak from the stomach, it would cause substantial damage to the outside cavity area. Chief cells produce pepsinogen. Pepsinogen is broken down to pepsin, which digests peptide bonds near aromatic amino acids, which shortens the peptide fragments. Parietal cells secrete hydrochloric acid, an intrinsic factor. These are the stomach acids we think of when we talk about stomach acid. G cells secrete gastrin. Gastrin induces the stomach to secrete more HCl and signals the stomach to contract, to mix the contents. After it travels through the stomach, it enters the duodenum, which is the first part of the small intestine. This is where most of the chemical digestion occurs in our body. There are different enzymes that are involved in breaking down and digesting our food. Disaccharides break down complex carbohydrates. Peptidases will break down proteins. Enteropeptidases activate trypsinogen and procarboxypeptidase. And secretin stimulates the release of pancreatic juices. Cholecystokinin stimulates bile release and satiety. If you have issues with this sort of secretion, you're going to notice eating disorders in which you never feel satiated. Certain diseases like Prader-Willi syndrome involve this issue. We've talked about ingestion and digestion, but let's go into some other accessory organs that are involved. The first one we need to go into is the pancreas, which releases pancreatic juices. This is very bicarbonate heavy and released into the small intestine. This is to help neutralize any residual acid from the stomach so that it doesn't damage the intestine as it travels down. Trypsinogen, chymotrypsinogen, carboxypeptidase A and B are also released here. Pancreatic lipase is also set out from the pancreas. Another key function of the pancreas is to release insulin and glucagon to the rest of the body to help with absorption or elevation of glucose levels. The liver largely produces bile, which is stored in the gallbladder or secreted into the duodenum. Bile, as a function, emulsifies fats and allows it to become soluble. If you remember seeing the micelles from a previous lecture, that's what bile helps to do. It processes nutrients as well, in the form of glycogenesis and glycogenolysis, and it stores and mobilizes fats. The liver is very important for detoxification, urea production, and gluconeogenesis as well. So we've gone into the first two portions. Let's go into the second portions, absorption and defecation, or egestion as it is also known. The jejunum and ileum continue the absorption of these nutrients in the form of villi, which increase the surface area to allow for more absorption. If you look at the lining of the small intestine, you're going to notice that it's lined like so. This is to increase the surface area to allow for more absorption to occur. Fat-soluble compounds will enter the lacteals from here, and water-soluble compounds will enter through the capillary beds. The large intestine absorbs water and salt along with forming semi-solid feces, which continue down until the rectum. Functions of the cecum, colon, and rectum are listed below. We'll finish our discussion of the digestive system there. As you can see, there's a very clear tract from start to finish, and you will need to know that for test day. We'll see you in the next lecture.